All right, in chapter 4, we're going to be talking about identifying the causes of cancer. And I'm going to have this split into two parts. In the first half, we'll talk about how researchers identify things that can cause cancer. And then in the second half, I'll discuss some of the factors that have been identified as causing cancer. And that second half is really kind of a lead up to some of the coming chapters in the book, which will individually go a little bit deeper into some of those different things that do cause cancer and talk in more depth about mechanism, etc. All right, so in terms of how we identify causes of cancer, there's really two main approaches, the epidemiological approach and the experimental approach. And I think as biologists, we tend to be more familiar with the experimental approach. If you're not familiar with epidemiology, that is the branch of medicine that basically studies frequency distribution of diseases in the human population. Um, and as we'll talk about a lot of times, it's really looking for correlation. So, what are things in the natural environment or in the human environment that are linked to disease? And of course, epidemiology also focuses on the spread of disease. So it has really come front and center, during the COVID-19 pandemic. We've relied a lot on epidemiologists. So, in terms of cancer, let's say epidemiologists are trying to do a study and they want to look at bladder cancer rates in two locations, Cleantown versus Chemtown. And they find that bladder cancer rates specifically are higher in Chemtown. Well, what does that mean? And the Chemtown picture always reminds me of the Lorax after they've cut down all the trees there. Let's say they say, okay, cancer rates are twice as high in Chemtown compared to Cleantown. Then the real question becomes, okay, how do we know if that difference is significant? Is it quote unquote real or not? Um, and some of this is going to, of course, come down to, well, how many people were they looking at in this study, right? If you say, oh, well, we had two cases of bladder cancer in Chemtown versus one in Cleantown, that's probably not going to be too convincing. If we look at their hypothetical data here, we say, oh, okay, well, they had 200 cases in per 100,000 people per year in Chemtown versus 100 in Cleantown. So that seems more substantial for sure. Um, but still, really, the question any scientist can ask is, okay, well, is that a statistically significant difference or not? So if you haven't taken a statistics class, I would definitely encourage you to do so. It'll come in, I think, very handy if you're pursuing science and medicine in any way. Um, but there's, of course, lots of different types of statistical tests you can do depending on the type of data that you're dealing with. But one of the most common one you've probably heard about is like p-value. So you could calculate a p-value, which is really estimating what is the probability that any difference between two measurements is just due to random chance, i.e., that there's really no effect of whatever the experimental variable is. Um, and you can you could sort of set a significant p-value of your choice, but most commonly you'll see p-values of like 0.05 being used to designate some statistical significance, and that would indicate that there's only a 5% chance that that difference would be due to random chance, and hence a 95% chance that that is a real statistically significant difference, in this case due to something different that's going on in Chemtown versus Cleantown. So for sure you're going to want some statistics, but even if you can show that difference is statistically significant, it's still possible there could be quite a few other explanations. Um, and this is always an issue in epidemiology, is there's quite a lot of different biases, for example. So there's what's called detection bias, publication bias, and experimenter bias. Um, let's just say detection bias is, well, what is the detection method they're using for bladder cancer and how many people are being screened. If you're just using, let's say, publicly available data, maybe people in Chemtown get screened for bladder cancer more regularly um, than in clean town. So that would be a detection bias. There's what are called confounding variables. We'll talk some more about those. Maybe people smoke more in Chemtown, and of course, smoking contributes to all kinds of cancer, including bladder cancer. Um, or it's still possible that this difference could be due to random fluctuation, right? Even with that 0.05 p-value, there's still a 5% chance that's just a random change. Um, so even with that statistical significance, just keep in mind there could be factors at play um, that that might mean, yeah, there's still really no true difference between those populations. So I just mentioned a confounding variable, and again, this is something that can be troublesome in lots of epidemiological studies, but we would define this as a variable that affects the risk of developing cancer in this case, and is in some way linked to the factor that's being investigated. So as we said, smoking would be a common confounding variable. If more people smoke in Chemtown, that is going to skew the data. Um, this has been a big problem in studies that have tried to look at the effect of alcohol on cancer rates because people that drink, people that spend more time in bars and establishments that sell alcohol are also more likely to smoke as well. So that may skew any data that's being collected there. In terms of cancer, age is another big confounding variable. Um, there's lots of interest in looking at the effect of prescription drugs on cancer, but of course, because elderly people are more likely to be taking prescription drugs, and we talked about the fact that age is really one of your greatest risk factors for developing cancer, again, that may skew that particular data set. So in terms of the types of epidemiological studies, there's really two main kinds I want to talk about: retrospective versus prospective studies. And retrospective, as that name would imply, is assessing past exposure. So looking at, okay, people that have already developed cancer in the past, and that's really a form of anecdotal data, right? Um, and so, in general, I would say retrospective studies are probably less powerful in terms of their predictive value than prospective, but there are all kinds of examples of very valuable retrospective studies. So, for example, these kind of studies were used to really cement the link between smoking and cancer. Um, one type of bias that creeps into retrospective studies is what's called recall bias. And this is really referring to the fact that, if you're relying on past data, past experience, for example, people that have developed cancer, and maybe you're questioning them about environmental exposures or something like that because they had cancer, they, may have spent a lot more time thinking about, oh, okay, well, what were things that I was exposed to that may have caused that, right? People are always trying to come up with these cause and effect links in their mind. So that can lead to this recall bias, as it's called. So on the flip side of that coin, prospective studies are going to actually follow people into the future, right, and see, okay, who develops cancer, who doesn't, going forward in time. And to do these properly, you really need a large study size, a large sample size, which makes them more challenging and more expensive to do. And of course, logistically, it's just more difficult to follow people going forward rather than collecting data that's already available. Um, so as I said, as we've seen, in COVID, epidemiology is a very important branch of science. Um, and it does have some big advantages, mainly that it's based on real life human experiences, right, compared to something that's happening in a laboratory situation or, in cell culture or something like that. Um, probably the biggest disadvantage, and this has really plagued epidemiology in a pretty large way, is that a lot of the findings tend to just be associations or correlations, and it can be really difficult to establish whether or not there's really a true cause and effect relationship, right? Going away from cancer, but back to COVID, we see this with, epidemiological studies looking at mask usage, right? Yeah, we see these correlations, but suggesting that mask wearing, face coverings are effective. But again, it's really hard to cement that down as true cause and effect. Um, and just to give you another example of sort of uh, an association that's really a bias, or in this case, it's something called a post hoc fallacy. You might be doing at a study looking at cancer rates, epidemiological study, and you find, oh, people who carry matches are have much more likely to develop cancer, have much higher cancer rates. Um, so, the human brain goes to cause and effect. Okay, well, our match is causing cancer, and of course, the answer is probably no, right? You have to dig a little deeper and realize that, okay, well, the reason people are carrying matches is because they're using them to light their cigarettes, and the cigarettes cause cancer, right? So, that's a somewhat silly and obvious example, but you could see cases where there might be things that are much more subtle, um, that could make it difficult to interpret the findings of an epidemiological study. So, given these issues, you may ask, okay, what makes epidemiological studies very believable then? We've got a short list of things you would want to look for. First being, yes, you want some statistical significance for sure. I would say a p-value of 0.05, as we talked about, or less. You'd want that 95% confidence interval, saying that, yeah, this difference, is due to some particular variable that we're looking at. And you would also want to see some evidence of a dose-response relationship, in particular if we're talking about, factors that cause cancer. Does a higher dose increase your cancer risk? And, we see this very clearly, for example, with cigarettes and lung cancer. So here you've got from basically no dose non-smokers to people who smoke less than 5 cigarettes a day, all the way up to, more than a pack a day. And for both men and women, we see a pretty linear increase in percent of individuals developing lung cancer, tied to increase in dose. So that's definitely going to make evidence more compelling if you can show that kind of relationship. Another thing you often hear about in epidemiological studies is magnitude, meaning what is your increased risk based on exposure to a particular factor. And I would say you probably want to see at least a twofold increase in risk for it to be something of concern. Just to give you an example, some of the lung cancer studies have shown that smoking, can increase your risk 25-fold. So that's very dramatic. But a lot of times you'll see numbers hovering, in that 1 to 3-fold increased risk rate. So I'd say you really want to look at things that are twofold or higher. And then lastly, you definitely want it to be repeatable. So you'd want to see multiple epidemiological studies showing the same effect. And this, I think, tends to be one problem with epidemiology, at least in the public eye or in the news cycle, is you'll have an individual study come out, suggesting, there's some link between, let's say, this environmental factor and a particular type of cancer. The media latches on to that one study, spreads that message broadly. People start panicking about that one factor, but then what you don't hear is, well, maybe 6 months later, a second study comes out that shows, maybe there is no link, right? Maybe there was some problem with that initial study, or maybe not even a problem, but just, something about the particular experimental design in that case showed a difference that we don't see with a slightly different design. So you'd really want to be in a situation where you've got multiple studies all pointing you in the same direction. And then, as we'll get into, for it to really be the most believable, you'd also want some support through experimental studies. And I'm not going to play this video here. I'll link to it out on Canvas, but it's just kind of a funny video uh, highlighting what I was just talking about, sort of the risks of trying to make lifestyle decisions based off single epidemiological studies. All right, so we have the epidemiological approach, and that is going to be complemented by experimental approach. And you know, you're probably familiar with experimental approaches that might be looking at things that could cause cancer, things like randomized trials, right? So how do cancer rates compare between groups that maybe receive different doses of a suspected carcinogen and people that receive no dose, for example, have a control group? These kinds of studies can be very powerful. But of course, you know, you're not going to treat humans with suspected carcinogens, right? That's going to be frowned upon. So in large part, this experimental approach, aside from like clinical trials of drugs and things like that, are going to rely on animal testing, especially when we're talking about causes of cancer. And when I say animal testing, usually this is going to be rats or mice. Um, so I really can't overemphasize the importance of animal testing in cancer research. And in fact, you know, it's law that any new chemical that's going to be introduced for human consumption, basically in any way, has to be tested on animals before it's going to be approved by the FDA. Um, and I just wanted to point out, in terms of talking about, you know, chemicals that cause cancer, various carcinogens, you know, one concern with animal testing as well, you know, animals are not humans, right? Mice are not exactly the same as humans. So maybe chemical that causes a specific type of cancer in mice may not cause that same type of cancer in humans. And there are some examples of that, but in general, I just wanted to point out here that carcinogens tend to cause the same types of cancers in you know, mice, rats, rabbits, as they do in humans, for the most part. So what that really means is we can have some confidence in these animal studies in terms of being able to apply what we learn there to humans and medicine. Uh, Now, of course, probably the biggest advantage of animal testing is that we can do these experiments under very controlled conditions, right? We can make sure the animals are genetically identical, or at least very uniform, and you know, have a nice control group, which adds a lot of weight to conclusions that we might be able to draw from these experiments. However, there are some disadvantages, as I just mentioned. You know, there are some physiological differences between mice, let's say, and humans. So what you learn in animal models may not always apply to the human situation. And of course, there's also ethical concerns, right? We're giving these poor animals cancer. But in general, you know, the the reasoning has been that these ethical concerns are outweighed by the potential benefit to reduce human death and suffering in the long run. Um, Another potential disadvantage to animal testing is that to do these animal studies, you usually have to treat the animals with higher doses of suspected carcinogens. And what humans would usually be exposed to, with the exception of maybe some, you know, workplace or like industrial high dose type exposures, most of the carcinogens we come into contact with are going to be very low dose. And so, you know, you could do these animal studies where you're treating with maybe 3 or 4 different high doses, right? Maybe you see something like this, where you get this nice dose response in the animal studies. It's very linear, and we would like to say, well, should we be able to extrapolate that back down to the low typical human dose or not. And in some cases, that may work out just fine. But as we'll talk about, there may be instances where it's not really a straight line between the low human dose and these higher animal doses, and so that estimation may be inaccurate. So why do these higher doses? Well, animal testing is expensive and it is time consuming. So typical animal experiment, using mice, think $1 million in three plus years. So that's going to be out of the reach of a lot of research groups. Um, so one alternative that's very commonly used because it's much less expensive is what's called the Ames test. And the Ames test utilizes bacteria instead of animals, and it is really a test for mutagens being things that mutate or change the DNA. And this is really using the assumption that things that mutate the DNA are also carcinogenic, and that is almost always the case. So if something alters your DNA, it usually has the potential to cause cancer. And so, in a nutshell, the way the Ames test works is they are using bacteria that have been intentionally mutated, either with a frame shift or point mutations, um, so that bacteria can no longer produce the amino acid histidine. Normal bacteria would be able to do that if supplied with an appropriate minimal growth media. So these bacteria, if plated on growth media lacking histidine, they simply cannot grow. And so what you do in this test is you take your potential carcinogen, the substance you want to test. Usually you mix it first with a liver homogenate to try to simulate the sort of metabolism that might happen to that substance in the animal or in the human's body. And you will then incubate that metabolized substance with bacteria on this minimal media. And the idea is if that substance or the metabolized version of that substance is a mutagen, it will start mutating the DNA in the bacteria. And at some frequency, you will basically reverse these mutations that were intentionally introduced so that now the bacteria can make its own histidine and can grow. And so the bottom line is this is really quantitative in that the more colonies, the more bacteria now that grow on these plates indicate the presence of a stronger mutagen in that original substance being tested there. So just counting up the colonies gives you an idea of the mutagenic potential of your particular substance in question. And again, that is tied in directly with carcinogenic potential in most cases. So here's an example of some different chemicals that have been tested in the Ames test. So you can see here, milligrams required to produce 100 colonies per plate. So you got something like aflatoxin that is very potent carcinogen and mutagen. And then on the other axis, you have increasing carcinogenic potential, basically ability to cause cancer in animals, right? And we see this pretty linear relationship here. So the increased mutagenic potency, according to the Ames test, correlates directly with increased cancer-causing ability in vivo. So Ames test is great, can be used to supplement animal models, animal testing, but of course, if we're worried about mice not being the same as humans, bacteria are definitely not the same as humans. But that being said, something that mutates bacterial DNA is also going to mutate animal DNA. So in that sense, it is a very strong test. However, there is a tendency for both false positives and false negatives in the Ames test, so you'd have to take that into account. And it's also worth noting that there are some chemicals that are not mutagens, so they wouldn't show up on the Ames test. as mutating DNA that can still cause cancer. So something like asbestos, for example. We'll talk about those mechanisms a little bit more later. All right, so we've talked about epidemiology, we've talked a little bit about experimental evidence. So what would we want if we wanted to really be able to claim, yep, this substance causes cancer? And the bottom line is the more sources of data that you've got, the better. Um, Ideally we'd want some data from the Ames test, and there's also some other lab tests that could be used to supplement the Ames test. We definitely want some data from animal testing, and we would like to see some epidemiological studies showing that, yeah, humans exposed to these agents out in the real world also have a higher risk of cancer. So if you have all three of those, that's painting a pretty vibrant picture that, yes, this particular substance is definitely linked to cancer. And, you'd feel more comfortable saying it can cause cancer. So I will stop there, and as I said, in the second half, we will talk about some of the different causes of cancer that have been identified from both epidemiological and animal testing.